Greetings,
Earthlings.
Today we are in for a whopper. Today we're going to do physiology of the urinary system. So first of all, I drew over here, that's right, today is a 2-board lecture. So I drew over here on this board the nephron. So this is the functional part of the kidney. And then I'm going to go through and change this picture and add a whole bunch of stuff and then tell you as we go so that we're doing sort of the anatomy and the physiology at the same time. So this is what a nephron really looks like. This is a picture from your book. So there's some of them that dip down barely at all into the medulla, and then there's some that go all the way down to the very tip, to the papillae of the pyramids, the papillae of the pyramids. So they kind of vary a little bit, and every single one, the blood vessel comes in, you have your afferent arterial, and then it comes into this little ball of yarn called the glomerulus, and then it goes up, and then it goes around. So all this purple is capillary. So this is exchange of gases and solutes and all sorts of things in between them before it goes into the collecting duct, and then it goes down into the calyces and then into the ureter. But to simplify, when we're talking about it, we take out all those blood vessels. So keep in mind that they're there, but this is what it's going to look like today as we kind of do a simplified version. And once again, it just shows you where it comes from in the kidney. So most of it stays all the way out here in the renal cortex, but they have some that their little loop goes all the way down into the medulla. You can follow along with us today. We have on page 176. Okay, so this whole thing is called the nephron. And like I said, this is blood vessels coming in all around and coming out in your afferent and efferent arterioles. So we're going to start with microscopic anatomy. After we erase the graffiti added by the Heyman family, lighting technician. There's a lot of light here, technician. Okay, so everything in orange here is the nephron. So that is the functional unit of the kidney. So it's the functional unit meaning it does the job of the kidney. So this is where it's split into three different parts, steps. Okay, so you put all three of these steps together and you get the function of the kidney. Okay, and when I say functional unit, we're talking about there are 1 million of these teeny tiny things per kidney. Okay. Okay, so we have glomerulus. Like I said, glomerulus, the word means ball of yarn, and it's a network of capillaries. Okay, so you have the afferent arterial here coming in, and then arterial. You have the efferent arterial leaving. And in between, you have the ball of capillaries, which is called the glomerulus. Okay? So it's got pores in between it that allow small things to squeeze out of the capillaries while large things stay in. Okay, so when we say small things, what are we talking about? What are small things that need to be filtered out into what will eventually become the urine? solutes and fluids, whereas large things are going to stay in. So large things would be like our red blood cells. They're too large that the whole cell isn't going to fit through these little pores. And proteins, like albumin, the big protein that helps with osmolarity, is too big to go through. So everything else is going to go out through the filter. So you imagine cleaning some apples off in your sieve thingy. What's that called? The apple stand, because they're big, and all of the solutes and the fluid and the water go through the pores. Okay, then you have your glomerular capsule. So this is a hollow cup-shaped structure that receives the filtrate. So at the very beginning of this nephron, where we have this yellow cup thing, this is going to be our glomerular capsule. Okay, so it's going to receive the filtrate. What is the filtrate? That is all the small things that's got that went through the filter is now filtrate. Okay, so anything that goes through the capillaries into the glomerular cup is going to come down, and now it's going to be filtrate. Okay, so there's some different layers here. So we have a parietal layer on the outside of this, which is simple squamous epithelium. Simple squamous is great for filtration, right? Then we have the visceral layer. This is stuck to the glomerulus, and it contains podocytes. So this is a type of cell that looks just like fingers clamped together. Okay, you see how there's holes in between it? It's just like that. And there's holes in between it. And those pores are what go through and make the filtrate. Okay, so here's a cool picture in our book. If you hit that ladder, your name is going to be mud. This is my son that is not going to hit the tripod ladder. Am I right? Cool picture in your book. Okay, so on page 985, this shows you what we're looking at, the capillary going into the glomerulus, and then if you take this and zoom in, then you have these cool little extensions of the podocytes, and in between them there's spaces, so we call those the filtration slits. Okay, so here's the fenestrations, the pores in the capillaries themselves, and then it needs to go through this parietal layer of the simple squamous, and then it needs to go through these filtration slits around the podocytes in order to become filtrate. So this is a fenestrated capillary, fenestrated endothelium. It needs to go through that, through the basal lamina of the glomerulus, and then through these filtration slits between the podocytes. Put all these three together, and that gives you the filtration membrane. So fluid and solutes come into the filtrate, and all the big stuff like red blood cells and albumin stay in the blood. The proximal convoluted tubules. So what a weird name, right? Proximal meaning closer to the point of attachment. So you think the proximal and distal end of my arm here, actually my upper limb. Okay, so proximal convoluted means all sorts of bent up and convoluted. Can't think of the word for that. And then a tubule. So right here You see how this one is closer to the glomerular capsule, so this is proximal to it, and this one is farther away. So this one is your proximal convoluted tubule, or PCT, and then over here, farther away, you have the distal convoluted tubule, because this is where it starts to get all crazy and convoluted again. Don't die now, marker. Okay, the proximal convoluted tubule is filled with cuboidal cells with microvilli. Okay, it has microvilli on it. That means it's going to be looking like shaggy when you look at it in a histology slide. And then it also has large microvilli. What does that tell us? If you have a lot of microvilli, then you need a lot of ATP or energy. So this tells us that it is active transport. So you're using ATP to push things where they don't want to go. Okay, so this is involved in reabsorption, one of those 3 steps to the function of nephrons that we talked about. Then we have the nephron loop. This used to be called the loop of Henle. That was the scientist that named it, but now we're trying to make it simpler for anatomy students. So I1 is the same thing. That's this big loop here. Okay, and like I said, some of them don't dip down very far at all and stay in the cortex of the kidney. Some of them go all the way to the bottom of the pyramid. So it has 2 different parts. It has the descending limb, okay, which is the thin part coming down here, the thin part, and the thicker part. So the thin part is simple squamous, and it's permeable to water, but not to salts. Okay, then the ascending limb that I forgot to write, so I just snuck it in there. That is this part coming up. So the ascending limb, the thick part is cuboidal, and this is permeable to salt, but not permeable to water. So that just tells us, okay, if different things are permeable at different times, then that means that those must be regulated separately. So now we'll get some salt coming out here. But not water. So the whole thing is the nephron loop. This is the ascending, because the filtrate is coming back up, and this is the descending because of the direction of the filtrate. Descending and ascending, the whole thing is the loop. Okay, then we have the distal convoluted tubule. Here we're still cuboidal like we were in the proximal, but we have no microvilli. So if there's no microvilli, then in a slide it would look clear and have a nice clear definition. Why am I bothering to go through and tell you what it looks like? If it's cuboidal, or if it's simple squamous, or if it has microvilli or not, you're thinking, why do we care? Because when you look at it in a histological slide, they are all in one slide together. Why? Why are these all in one slide together? Focus. Because remember that we said there's millions and millions of these in a kidney. So if you cut across the kidney and make it into a slide, there's a whole bunch all next to each other. So you might get this glomerulus, but it might be right next to a distal convoluted tubule of the nephron next to it, or it might be next to a collecting duct. In this slide, they all get stuck together. So how do you tell who is who? Okay, this is obviously a capsule. So around the capsule is going to be your glomerulus. And then out here you just have a bunch of tubules. And the way that you can tell is just by looking at it, does it have microvilli or not? So you see how this one's nice and fuzzy, this one's nice and fuzzy, but this one has a nice clear lumen that you can see. So that means this is a distal convoluted tubule and this is a fuzzy proximal convoluted tubule. So it just tells you what you're looking at. Then at the end we have our collecting duct. Why is it called a collecting duct? Because it's going to take several the nephrons are going to dump into one collecting duct. That's what this little cutoff part is another distal convoluted tubule attaching to it. It's going to take all of them and take all the filtrate and filter down, shouldn't use the word filter, and run down into the calyces, the minor and then the major, right? And then the pelvis, and then to the ureter. Receives filtrate from many, not neurons, nephrons. Okay, they run through the pyramids and enter into the minor calyces. It's made up of two different types of cells, the principal cells and the intercalated cells. So the principal cells maintain the body's water and sodium balance. So these have receptors on it for ADH and for aldosterone. The principal cells do of the collecting duct. ADH is antidiuretic hormone, which you remember from when we talked about the endocrine system. Antidiuretic hormone controls the water balance. And then aldosterone has receptors on it. It has aldosterone receptors, which is another hormone that's going to reabsorb sodium and secrete potassium. So once again, keeping those specific ions in check. The other cells that make up the collecting duct are called intercalated cells, and these maintain the acid-base balance of the body, which is done by either collecting all of those hydrogens, which would leave less hydrogen in the body, right? That's going to make it alkaline, versus secreting more of those hydrogens. Increasing the number of hydrogens that you have is going to change the pH of the body and make it more acidic. So like I said, the first way, the quickest way, is to hold your breath or to hyperventilate so your respiratory system can do it. And the second way is your kidneys can either secrete or reabsorb those hydrogens, and that's how your body maintains its pH balance. So we talked about the different types of nephrons, but it's always good to throw in some terminology. So we have the cortical nephrons, which are about 85%. These are the ones that are entirely in the cortex. Some don't even have loops. I know. It's shocking. It's shocking to think about. Then we have juxtamedullary nephrons. Juxta meaning next to or against. Against the medulla, these are the ones with the long loop, and they work to concentrate urine. Are you drinking enough? Are you keeping yourself well hydrated so that your juxtamedullary nephrons have plenty to do? Or maybe not a lot to do, because they can just let it go into the filtrate. Okay, and then last but not least, the last anatomical thing that we need to throw in there before we talk about the function is the juxtaglomerular complex. So this is a complex because it's like a little chunk of cells, and it's next to the glomerulus. Okay, so right here is the glomerulus, and the afferent arteriole, which is right next to it, and the ascending limb. And I know you're like, wait a minute, the ascending limb is way over here, but you got to think of them all crammed in there together. So this ascending limb up against the glomerulus next to it have these complexes, and they have these three different types of cell that help to make modifications to the filtrate. Why would you want to make modifications to the filtrate? Homeostasis. You're constantly looking at the balance of all sorts of different solutes and all sorts of different ions and all sorts of different gases. So you have lots of different cells that are monitoring different things. Okay, the macula densa, these are cells with chemoreceptors, so they're looking at chemical content, and they're monitoring sodium chloride, so salt content. So it's like, oh no, she had pretzels again for lunch. So her blood is just streaming with all this salt. Okay, we need to look into that. Granular cells are cells with mechanoreceptors that are looking at blood pressure. Why would your kidneys care about blood pressure? Because that's how you increase or decrease blood pressure, right? Well, that's one of the things that affect it, is how much fluid or how little fluid you have in your vessels. Okay, so these can constrict arterioles, or they can also release renin. Renin is a hormone that's going to be involved in blood pressure and the amount of plasma, therefore the amount of fluid in your body. And then last but not least, we have mesangial cells, which are the walkie-talkie operators between these two types of cells to keep them all in the right place. Here is a beautiful picture of your book. Okay, so all the purple are these complexes that we're looking at. And like I said, they're there kind of stuck in between everything else, working on the concentrations of specific things. That's on page 983 in the book. Step one. So in physiology, we have three major processes: filtration, reabsorption, secretion. We're going to start off with filtration. So this is basically the process of making the glomerular filtrate. So coming out of the glomerular capillaries into the capsule and making filtrate. Okay, it's filtered at this point by size. Throughout this process, it's filtered according to different things, or secreted or reabsorbed according to different properties. Here it is by size. It's basically like when you clean out your fridge. First thing you're going to do is just take everything out of your fridge, right? And then you're going to mess with a bunch of things and then decide exactly what you want in it and what you don't want in it. So this is the step where you take out everything. Anything that's too big, bigger than 5 nanometers, nanometers is small, stays in the blood, because you don't need to filter out your blood proteins, you don't need to filter out your red blood cells, your white blood cells, or your platelets. It's the solutes and the little ions you need to worry about. Okay? And the filtration rate is determined by pressures. Remember, we were talking about pressures in the blood and the pressures that oppose the capillaries. Same thing here, because these are capillaries, right? Okay, so you have forces that are pushing the solutes into the capsule. It's pushing it out of that capillary. Hydrostatic pressure, which is basically like the blood pressure in the capillaries, is pushing it out. Hydrostatic pressure of a glomerular capsule equals 5 millimeters of mercury. Then you have some things that are opposing it, that are stopping the blood from flowing out of those capillaries and being filtered into filtrate. This includes the hydrostatic pressure in the capsular space. So the capsular space also has blood pressure in it, right? Interstitial fluid. So it's pushing against it at 30 millimeters of mercury. Then you also have some osmotic pressure called colloid osmotic pressure in the capsule itself. And this is water wanting to be with those big proteins. So water likes to follow salt, and it likes to follow solutes. So if you've got those great big blood proteins, albumin, water wants to be with them. So that's another force that's opposing it coming out. So if you have 55 millimeters pushing it out and 30 plus 15 millimeters of mercury opposing it coming out, then you get an average of 10 millimeters of mercury, which is the net pressure out. So in our beautiful picture, step 1, green, feel like a green. Step 1 is filtration. I know how to draw arrows. So you have a net of 10 millimeters of mercury pushing all of these things out of the capsule, out into the capsule, making it filtrate. based on its size. The glomerular filtration rate is the total volume of filtrate formed per minute, but per minute we're talking about all of glomeruli. Glomeruli is plural for glomerulus, so we're looking at the total filtration rate of all the glomeruli per minute. It's directly proportional to the net filtration rate. So these are three things that can change the filtration rate. The first one is you alter the amount of surface for filtration. So that's not going to happen very often, right? Maybe if you donate a kidney to your brother. But most of the time you're going to have the same amount of filtration surface area. The second one is to filter the permeability of the membrane itself, make it more or less permeable to water or to salts based on what's going on in your body at the time. The third one is to alter the net filtration pressure. Okay, this is done by changing the diameter of the arteriole. So as you change the diameter, you're changing the pressure and therefore changing the rate. Let's look at a picture, shall we? Okay, so this shows you under normal circumstances, you have a net filtration pressure of about 10. But if you start to constrict the afferent, see how the efferent is the same in all these scenarios? Okay, then it's going to be less than 10. You're going to get a slower rate of filtration. If you dilate that afferent arteriole, then you're going to get a greater net pressure and therefore a faster rate. Okay. Focus. There, now you can read it. Okay, so how do we control this? There's intrinsic controls and extrinsic controls. Intrinsic are the ones that you're using right now. So for most of the time in everyday normal life, it's going to be renal autoregulation. So the kidneys are going to take care of everything all by themselves and figure out what they need. And you don't need any input from anywhere else. And it's going to do that mostly, like I said, by changing the diameter of the arterioles. Good thing you have all that smooth muscle and arteries, huh? And the sphincters to turn on or off an entire capillary bed. Okay, and then we have the extrinsic controls. So these are under extreme circumstances. If your blood pressure suddenly drops, if you're hemorrhaging, that's an extreme situation where you have less than 80 millimeters of mercury as your blood pressure, then things are looking grim, okay? And then all of a sudden, extrinsic controls are things outside of the kidneys can now help to control that filtration, okay? Nervous system or endocrine system, because those are the two systems that control everything. Sympathetic nervous system, fight or flight. It's going to release the hormone norepinephrine, which constricts all of the afferent arterioles. Okay, something else that can help is the endocrine system. So angiotensinogen can be converted into angiotensin 1 and then angiotensin 2, both of these hormones. And it does that by the enzyme renin. Okay, so more or less renin that you make is going to change that pathway of that endocrine pathway. Okay, so what change is this going to cause? Causes changes all over the body. So the first one is it going to constrict the arteries throughout the body. It's going to activate the thirst center of the hypothalamus and give you this huge craving for some fluids. The kidneys are going to be told to reabsorb sodium because water is going to follow salt. You're going to increase the fluid reabsorption as the efferent arterioles constrict, and you're going to decrease the total surface area in your glomerular capillaries. So you can change the capillary surface area. So you put it all together, and what's it going to do? Increase your blood pressure. So they all work together to increase your blood pressure and to take care of you if you have a major hemorrhage. In extreme circumstances, there's lots of things that your body can do and lots of things that can change to get that blood pressure back up again.